Welcome to Edutube. Science is simple. In this video, we'll explore how to test for different food molecules. Before starting any test, it's important to prepare the food sample properly. If the food is solid, begin by crushing or grinding it using a mortar and pestle. Then, mix the crushed food with a small amount of distilled water and stir it well to make a liquid mixture. If necessary, filter the mixture to remove any large particles. This helps the test reagents react more effectively. If the food is already a liquid, you can use it directly for testing. To test for reducing sugars, like glucose, maltose, or fructose, we use Benedict's test. Reducing sugars are carbohydrates that can donate electrons to the copper ions in Benedict's solution. Start by placing 1 milliliter of the food solution into a test tube. Add 2 milliliters of Benedict's solution, which is blue in color. Then place the test tube in a hot water bath at around 80 degrees Celsius for about 5 minutes. If no reducing sugar is present, the solution will remain blue. But if reducing sugar is present, the solution will change color, starting from green, then turning yellow, orange, and finally a brick red precipitate, depending on how much sugar is in the sample. The stronger the color change, the more sugar is present. Iodine test is a simple and quick way to check if a food sample contains starch, a complex carbohydrate found in foods like rice, potatoes, and bread. To perform this test, we use iodine solution, which is yellow-brown in color. Start by placing 1 milliliter of the food solution into a test tube. Then, add a few drops of iodine solution. If starch is present, the iodine will react with it, and the mixture will turn a blue-black color. If there is no starch, the solution will remain its original yellow-brown color. Be sure to handle iodine carefully, as it can stain your skin and clothing. Next, we have the Biuret test, which is used to detect proteins. The Biuret solution contains sodium hydroxide and copper 2 sulfate. When added to a solution containing protein, these chemicals react with the peptide bonds, causing a color change. To carry out the test, add 1 milliliter of the liquid food sample into a test tube. Then add 2 milliliters of Biuret solution. If protein is present, the mixture will turn from blue to purple or lilac. If no protein is present, the solution will remain blue. Finally, let's look at the emulsion test for lipids. If the food sample is solid, like nuts, cheese, or butter, it should first be crushed to increase the surface area, then mixed with ethanol to dissolve the lipids. If the food is already a liquid, like oil or milk, you can simply shake it with ethanol. After that, pour the mixture into another test tube containing water. If lipids are present, a cloudy white layer, known as an emulsion, will form at the top of the solution. This happens because lipids dissolve in ethanol but not in water. If there are no lipids, the mixture will remain clear. In summary, the reducing sugar test involves adding Benedict's solution to the food sample, heating the mixture, and observing any color change. A color shift from blue to green, yellow, orange, or brick red indicates the presence of reducing sugars. For the starch test, simply add iodine solution to the sample. If it turns blue-black, starch is present. The Biuret test is used to detect proteins. After adding sodium hydroxide and copper 2 sulfate to the food sample, a purple or lilac color shows that protein is present. Lastly, the emulsion test checks for fats. When the food sample is mixed with ethanol and then added to water, the presence of lipids is indicated by a cloudy white layer forming at the top. If this video helped you understand food tests better, make sure to like, subscribe, and hit that bell icon so you never miss a bite-sized science lesson from Edutube. Until next time, keep learning, stay curious, and remember, science is simple.